Copelli. So he'll be talking about collective neuronal behavior and the critical brain hypothesis.
Okay, thank you. Good morning, everyone. Thank you for being here again. So, as I told you yesterday, I was trying to paint this panoramic view of this field from the last 20 years or so. And today, I hope to arrive at the present of this field, or at least my view of this field, okay? So, let me briefly review what I told you yesterday, telling the story about this paper. So, in this 2019 paper, we took urethane data, so anesthetized animals, and studied avalanches, and we saw these scaling laws for size and duration of avalanches, obtained these exponents tau and tau t, and then obtained this third exponent, the combination of exponents here, and showed that borrowing results from statistical mechanics, we know that they are not independent. They should satisfy the scaling relation, and this is a very high bar to pass. It's very hard to find a model which is not critical and satisfies these relations. And since the two sides of this equation depends on the level of synchronization in the data of the CV, the coefficient of variation of the collective population spiking activity, which is here, we find this crossing here, and we interpreted this as a sign of a phase transition because here you have the scaling relation satisfied. And when you look at this picture, the natural thing to do is to interpret this thing here as some kind of order parameter, control parameter, a control parameter of the brain, which you're not controlling because it's self-organized. It's going here back and forth automatically because of urethane anesthesia. And given that this is the residence time of all animals, the group data, so this you interpret that, okay, the system is critical, but en passant, right? Just in passing, because it goes out of criticality here and here, here. Right, so this is, and moreover, the exponents are non-trivial here, which would require a different model than the one people have been used to. And then, a couple of years later, we showed, well, starting with Tawan Carvalho, this brilliant student of mine, who proposed that actually you can just use a trivial model and introduce subsampling in the model, which happens in the data, and this happens to distort the exponents, so they become apparently non-trivial. And so this is it's a simpler explanation for the data. But this thing works only if this model is very close to the critical point. You're allowed to cut some slack, like 3% off the criticality, but this changes the picture of interpretation of this data because the model can generate, with a very mild change of parameters, this whole distribution of CV that you observe in the data. So this wide distribution of CV here is obtained with a very minor change in parameter around the critical point. So if this is correct, then the interpretation of CV as being a control parameter is wrong. We were wrong. And because this is a true control parameter, it's a model. That's the advantage of having a model. It can guide you somehow. And according to this interpretation, then, it's not criticality en passant, but rather all the time, near criticality. So it's a stronger result. But, of course, it's based on a model. Wow, a model suggested that. Is it true? How could you probe this idea just looking at the data without a model? I'm all for models, okay? But there's only so much the models can tell you. And then, so that's where this third paper comes in, led by Daniel Castro. And in order to tell you guys what he did, I have to pause a little bit and talk about the real normalization group. So the real normalization group was developed in the second half of the 20th century and eventually led to the Nobel Prize by Wilson here 1982, I think. And what is it about? Consider this model. That's the Ising model, okay, for ferromagnetism. You have spins which can be up or down, and you have some coupling J naught among them, and you have some Hamiltonian, some energy function that describes, from which you can calculate the statistical mechanics and do all the calculations. Well, depending on the temperature, these spins here will try to align somehow. So the renormalization group is a mathematical procedure that you do on a model, which looks like this. So, oops. I don't know what I have to do. Well, I just got rid of it. Oh. I'm not sure I'm speaking English, actually. It's broken English. Well, anyway, so you do this mathematical procedure by which you take, for instance, this group of spins, and you take some kind of average, and you transform this group of spins like in a super spin, which is like looking at those spins from a different scale. I mean, you do this mathematically, okay? There's some calculation to be done. And those group of spin blocks now are coupled by some different coupling J1, which is a function of this other one and a function of the previous temperature. So you map the temperature here to some temperature here, you map this coupling J0 to this coupling after the first iteration of the renormalization group. And then, guess what? You do it again. So you change scale again, and you transform these groups of spins into super spins, and this coupling J1 into coupling J2, and so on and so forth. And you keep doing this transformation. So as you transform these couplings according to this procedure, what you're doing is changing the scale of your system. And if and when in these dynamics, this procedure leads to a fixed point in the couplings parameter, that means you've reached a point where you have scale invariance by definition, because you keep changing the scale of the system, and things stop changing. So you have scale invariance, which is a signature of a critical point. Okay? So this, what I showed you before, was a cartoon. This is from a review by Michael Fisher, in which he shows, well, tries to show this. They have this plane of temperature control parameter and external field. You have a critical point here, and you change these parameters according to this procedure, and on and on and on and on, and eventually you reach some kind of fixed point here, which is a separatrix between what would be a supercritical system and a supercritical system on the two sides. So for those familiar with linear dynamics, it's like a saddle point which separates two different attractors, completely ordered and completely disordered. And the critical point is right in between. And it turns out that in this mathematical procedure, this appears as some kind of separatrix here, this critical point, okay? So at this critical point, because of its very special nature, is it okay? Yeah? Tudo bem? Okay. Should I keep trying? Yeah? Because of the special nature of this point, what you expect here from the theory, I'm just talking about theory now, okay? Theory in models, theoretical models. Here, when you renormalize the activity of whatever you're looking at, whatever is the natural observable in your system, here, after you do all these procedures, it will show scale invariant features and non-Gaussian distributions, all signatures of non-triviality. It's a very non-trivial point. Here it becomes trivial, here it becomes trivial. Completely ordered, completely disordered. And here it becomes, had some special signatures of non-triviality, right? Which is associated with this critical point. So this is the gold standard in theoretical physics for understanding phase transitions in models. In models, which will be a dead end for us because we don't have a good model of the brain, by no means, right? I have sketches and rough models for here and there. But, fortunately, then in 2019, Bill Bialek introduced the phenomenological renormalization group, which borrows these ideas from the renormalization group and tries to get rid of the model and use just the data. So it's data-driven, model-free. So these ideas were introduced in this very nice paper, 2017, Journal of Statistical Physics, and then later on in PRL by Michelin and Bialek's group in 2019. So this second paper is in the Google Drive folder, okay? If you want to have a read. Bill writes very well, and so you should read his papers. And he also is a very excellent gives a lecture. So this is fairly recent, and there's a couple of papers here. I'm going to talk about this one here and about the next one in the sequence. So what is the idea here? We don't have a model. That means we don't have the interactions. We don't know when you take a measurement of neurons, very rarely you know who is interacting to whom and how. Excitatory connection, inhibitory connection, whatever. So this is data from Bill's paper. This is calcium imaging from the hippocampus, I think. So instead of, remember you have the spins, I group neighboring spins, because neighboring spins are interacting. I know because it's my model, we know what's going on. Here we don't know. So not knowing the interaction, you take a proxy of interaction, which is correlation. You could use other things as well, okay? You could do maximum mutual information, blah, 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 and whatnot. But in the simplest case, just correlation. So you take the most correlated pair and you group them together. Just sum the activity and eventually can normalize the activity or not. It doesn't really matter here. And then the other two most correlated pairs and sum them here and do this thing. And then you have a group of, say, superneurons, a cluster of 2 neurons. And then you redo it again, you have a cluster of 4 neurons. And then clusters of 8 neurons, etc., etc., etc. So you are renormalizing your collective activity in the spirit of the renormalization group. And then you observe whether you check whether as you do this, as your cluster size increases, you check whether observables scale non-trivially with cluster size, and whether this normalized activity converges to something that looks non-Gaussian. Look, most of you guys are physicists, so you heard about the central limit theorem. You sum random variables, you expect the variance to scale linearly with your system size, right? Given the assumptions of the central limit theorem, which is they don't have correlations. If they do, if they are correlated, you expect your variance to increase with the square of your system size. For instance, what happens here? We'll see in a minute. So this is what we've been calling, yeah?
Sorry, one question, just to be sure if I understood. So when you group neurons together, you just sum up the activity? Just sum.
Yes, just sum, yeah. And we will do it with spikes. People have done it here. I think this is Poncelvers. They've done it with fMRI. It's pretty general. You can tweak a little bit with the procedure, but this part is simple. Just sum it. So I'm going to call it this real space phenomenological renormalization group, as opposed to momentum space, which is something that if you've taken a course on phase transitions and the renormalization group, you've heard about momentum space. And I'll talk about it a little bit later, okay? And so what is it that they propose to look at? For instance, the probability of silence as you go from here to here to here as a function of cluster size. You can imagine that the more you cluster together this activity, the less probable it is that you're going to have silence in this time series. And this is minus log of P of silence. And for instance, and it scales as exponential of minus some constants, cluster size to the power beta. When beta equals 1, trust me, it's a Poisson process. It's a trivial process. If beta is not 1, you have something non-trivial there. I'm going to focus a lot on this normalized activity. As you increase cluster size, it's converging to something clearly not Gaussian. Okay? This is really non-Gaussian. And even though Bill didn't do this here, if you shuffle everything and destroy the correlation, then it becomes Gaussian. Okay, so there's always the control, of course. The variance that I mentioned before, if you just sum up, calculate the variance of these clusters here, you see that these two dashed lines, this is exponent 1, exponent 2. The exponent here is alpha. So the variance increases with cluster size with some non-trivial exponent between 1 and 2. So 1 and 2 will be trivial in different ways, but both trivial. And exponent z, I'm sorry, this is exponent beta, this has no exponent here. This is exponent alpha, exponent z is how the autocorrelation time of this time series increases with cluster size. So the trivial case here, you increase, if these things were uncorrelated, you have this correlation falling all with the same characteristic time. So trivial case is z equals 0. But that's not what you find here. You find something here. And finally, the hardest of all, at least harder to interpret with my hands like this, is you look at the eigenvalue spectrum of the covariance matrix. So you have rank of the eigenvalues of the covariance matrix, and here the value of the eigenvalue. And you also find a power law here with some exponent mu. I will not delve much into this. Just so you know that he has chosen, for instance, these 4 exponents to have a look at. These 3 have trivial values to compare with. And this one, if it's trivial, it doesn't even have an exponent here to start with. Right. So this is what he obtained. It's a very nice paper. So we decided to apply these ideas to this urethane data. Again, anesthetized animals. So I've shown you this before. So we have the coefficient variation, so the level synchronization of the spikes in the primary visual cortex from very asynchronous behavior to very synchronous behavior as measured by CV. And we parse the data by CV and run this PRG analysis on it and see what happens. Okay? So again, I'm showing you one instance of CV as a function of time. Low CV is asynchronous. High CV is more synchronous behavior. Intermediate CV is something in between. And then we run this procedure. So what I'm going to show now is probably the most important figure of this paper, is the normalized activity of these spikes after you do all the interactions, as many as you can. Of course, if at each interaction you divide by 2 the number of neurons, you need many neurons to start with, okay? So we have at least 128 neurons, which is 2 to the power 7, I think, right? So we can run at least 6 iterations of the procedure. And what you do find is, look, color-coded here is the value of the synchronization in the original data. So you see here that if activity is sufficiently asynchronous, that CV is as low as 0.5, when you renormalize activity, you get something close to a Gaussian. This is the log linear scale here. Whereas for high CV, so highly synchronized data, you have something that's clearly non-Gaussian. So apparently, the data is showing signs of criticality depending on how synchronized it is. If the natural plot to do is not this one, the natural plot to do is the next one, which is some measurement of non-Gaussianity as a function of CV, right? So the measure of non-Gaussianity which we chose is the simplest one you can think of, which is the kurtosis of the distribution, okay? So kurtosis, we learned statistical course, is just like the x, something like that, divided by x squared, for instance, okay? So for Gaussian, I think this is 3 or something, some small value. If it's non-Gaussian, then this value can increase and can decrease, but for these distributions, it will increase. So this is what you get. This is kurtosis as a function of this value of CV, and it's very interesting because it keeps a low value here. This gray bar here is what you get for shuffle data, okay? So this is control. You shuffle your spikes, you get nothing. Just Gaussians, 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 and low kurtosis. And for the data, you have this thing here, which is comparable to the surrogate data, so we count this as not heavy scaling, and then eventually this thing takes off. So you have signs of non-trivial activity here. So if we use this as a criterion, and then you have scaling here and no scaling there, okay? Here is an animated GIF that only students can do. I don't know how to do this thing, but so CV is varying along the time series, and here you can see the randomized activity after 5, 6 iterations. Okay, you see when CV is high, you have something which is highly non-Gaussian. When CV is low, it's very close to a Gaussian distribution. Okay? So, nice. So, what this analysis is telling us is that CV is indeed a poor control parameter, as predicted by the model that I showed you yesterday. Why is it so? Because whenever this kurtosis is high enough, this is a signature by the phenomenological renormalization group technique of non-trivial scaling. So we have non-trivial scaling everywhere here in this whole range of CV. And not just in that very intermediate part that we showed yesterday, according to avalanche analysis, which, as I commented, has several methodological issues. So they appear, and they are confronted here, and this gives you a different answer, which I find nice. So my current understanding is that this technique introduced by Bialek solves or bypasses issues that appear in this avalanche analysis. Okay? Circumvents these issues. And by the way, as I've shown you yesterday, in a cartoon plot of a second-order phase transition in which you have a control parameter here, say, in physics it will be, I don't know, the temperature, and here you have some order parameter. In that toy model that I showed you, you have some coupling between excitatory coupling, and eventually here, this will be the density of active sites. You have something like this, right? That's the cartoon of a second-order phase transition, and that's the critical point. You have to be very careful not to be misguided and think that this is such a plot, because it is not. It looks like this one, but it is a completely different plot, because here, this is not a control parameter. This is a sign, the larger the kappa, the less trivial your activity is, and the closer you are, I think, to a critical point. So everything here is critical, okay? It's not like this one. So this plot looks like this one, but it's not. And as soon as kurtosis stabilizes here departs from surrogate values, so kurtosis here is the vertical axis, and here is the horizontal axis, okay? Please notice that. So this is surrogate levels. As soon as you depart from surrogate levels, the values of those exponents that I mentioned before, alpha, beta, mu, z, so how variance scales with cluster size, how the probability of silence scale with cluster size, or the log of the probability of silence, how the eigenvalue spectrum scale with cluster size, and, no, scale with rank, I'm sorry, and how characteristic autocorrelation time scales with cluster size. So the values of exponents more or less stabilize after you depart here. So the exponents are stable, and therefore pointing to, okay, you are the same, several quotes, critical point, somehow, okay? Cool. And just,
Mauro,
yeah.
Sorry. Just in the last slide. So when you look at the CV, the high CV, right?
Yeah.
It looks a little bit more like a periodic kind of activity, no? So I wonder, so my question is that if you do this analysis on something that is periodic, would you get something that, because probably is non-Gaussian also, right? Because it's periodic. So I'm just wondering if you would get something very similar to what you're getting here, although it's not really scale free or something like that.
The system in high CV is very synchronized, but not periodic. So it's what is called synchronous aperiodic. There's some fancy name for that. I forgot the name, but so they go up together, they go down, but they don't do this regularly. It's synchronous irregular. That's the name, I think, yeah.
Can you remind again of how exactly you define CV here? The way I know it is that CV of 1 is a Poissonian process, and
Yes,
CV below
that's true.
1 is
CV
regular.
of 1 is a Poisson, that's right. So CV1 is a Poisson process, but depends on CV of what, right? So
Yeah.
CV
Sigma over mu,
is
right?
just standard deviation divided by the mean. The
Yeah.
question is of what? So this is true, what you're saying is true for a Poisson process. Probability of interspike interval, right? Which for a Poisson is exponential, and therefore this is true. But here we're talking about the CV of the population firing rate. So it's completely different. It has no obligation of being. So whenever CV is 1 here, I cannot compare with a Poisson process. I'm just talking about a summed activity of this. These
But it would
spikes.
still be true that if the CV is much smaller than 1, that sigma is smaller than mu, so you'd have a tight distribution of your population activity. So it
Yes.
would have to be somewhat periodic
Periodic?
for small CVs. Yeah.
No, for small CV you have this asynchronous irregular activity. So your firing rate is fluctuating not very much around the mean. In what this Sompolinsky paper from the 80s will call a chaotic system, because it was fashionable by then to call everything chaotic, but it's just irregular asynchronous activity, right? So, but I'm used to that question because it's very common, because we tend to look at this quantity mostly in single spikes, right? In single neuron spiking, yeah. But it's different. Right, so let me go back here. No, we've been here before. Okay, so here I'm just showing that these exponents have more or less stable values across different animals. I had 9 animals, I think, in this cohort. And this is interesting. If you do the data for, if you run this kurtosis function for different animals, so these are the names of the animals, you see that they depart from these trivial values here around the same value, around 0.7, something like that. And this is very consistent among animals, the way where they depart, despite the fact that the probability of finding a given CV in a different animal is completely different. So you anesthetize with urethane, and each animal has its own history in terms of how synchronization will evolve in time. Some get more synchronous than not. If they're too asynchronous for too long, you probably got it wrong, the anesthesia wrong. You should stop your experiment. So this has been taken care of, okay, to avoid this dangerous outlier, this bad outlier. But so you see that even though this is the way they behave is very different, the way they depart here is very similar. And if you take this some kind of criterion as, okay, it is critical here, it's not critical there, then you can calculate stuff like this. Okay, so what is the probability of finding an animal in this scaling region as opposed to this non-scaling region, or in the region where some animals can be here, so below 0.7, and still have some signs of criticality. So you can run some basic statistics on that. It's very simple to define. You have the probability of find scaling, which is this darker color here, as a function of CV. So this is the group data. And this is the probability density of finding no scaling as a function of CV. You see, you have some coexistent region here, where some animals can and some animals won't present scaling in this range. And overall, you find that scaling occurs 76% of the time for this data set. You can do this analysis also on each animal, and you find that the average across animals is about the same here. You can do the probability of find scaling given some CV, and it gives you a curve like this, which increases and saturates to 1 around here. I don't know, there's a characteristic. The half height value is, again, 0.7. Okay, so the conclusion of this thing is that if we accept that non-trivial scaling is a good signature of criticality, then the cortex under urethane goes in and out of criticality and stays in criticality most of the time, like 76% of the time for this data set. And therefore, this data supports the prediction of the model that we had before, the second model, which is very nice. I would not have expected that this model had any right to predict such a thing, but apparently it did, in the sense that it predicts, okay, you have to be close to the critical point. That's the extent to which the model was correct. So there's a wide range of synchronous high CV states, a manifestation of a single critical point. There's some caveats for the technique. So there's no time domain normalization. The issue of how you bin the data and at which time scale you look at this phenomena is an issue in this field, and I'm not going to discuss this because it's too long and uncharted territory to some extent. And no information about the universality class, because no matter how beautiful I find this method, the exponents it gives us are not related to the bona fide critical exponents defining in statistical mechanics course. Okay, so there's this disconnection there. And the thing is, the phenomenological normalization group on non-anesthetized mice, which has been done by Miguel Munoz's group in Granada, does show signals of criticality in states which are clearly asynchronous. If the animal is awake, what you obtain is low CV. Here, low CV means non-criticality. There, CV doesn't matter because it's always low, and it shows signal criticality. So, apparently, not all asynchronous states are alike. They look very much the same. That's the reason why urethane got some attention when it was introduced, because it was considered a mimic of something that goes very asynchronous and very synchronous. This very asynchronous looking like something like REM sleep or something, which electrophysiologically looks a little bit like waking state, and very synchronous, like slow wave sleep. So, okay. Let me go back to Italo's question. The way I started these lectures was with a hand wave argument that criticality, the promise, would be that a system in a critical state has several functional advantages. Then I go and show you data from experiments in slices, and anesthetized animals, which are doing nothing, right? So, well, the closest I think we have gotten to this link between brain criticality and some functional advantages, again, is not very strong. Remember, I showed yesterday we had predicted that dynamic range should be maximized at the critical point, and they do obtain something like this, like stimulating a slice. So the slice responds optimally at the critical point of the slice. Okay, but it's still a slice, right? And they did also for the anesthetized animals. So yes, the somatosensory cortex of the rats respond optimally near a critical point. Fine. I like these results. But still, the question Italo asked yesterday is a question that should be reverberating all the time. And it is in this community, except that people didn't know how to address it. I think we have addressed. So I'm going to show you a clear example, okay? Can brain criticality be linked to enhanced performance in a task? So this is as objective as it can get, I think. There's a task, and there's performance, and can we make a link with brain criticality? So my claim is that we have shown that the answer is yes. So now we have the brain, Allen Brain Observatory experiment, view behavior neuropixels. So this, for those who are not in neuroscience or who have never heard about this institution, they perform massive neuroscience experiments and make the data publicly available. It's fantastic. It's a massive data set. And so here, we're talking about mice which have, having pre-trained, and they have their head fixed. They are on a carousel of sorts. They can move freely like this. And their task is to detect change of images in a sequence. So they are presented with an image and another image and another image, and then eventually this image may change or not. If the image changes, they have to lick to show that they have noticed a change of image and get rewarded, right? And they have neuropixels implanted everywhere. I'm just going to look at primary visual cortex with recordings of primary visual cortex with at least 128 units, so I can apply Bialec's phenomenological renormalization group to this data. Okay, so when you do this filtering here, you end up with 62 sessions by 44 mice, which is a lot. So how do I assess criticality in this data set? Well, so let me, again, remind you that, okay, so they're recording in V1 here. So initially, you have this active phase of the experiment. So for 60 minutes, the mouse is watching these pictures and having to lick or not to detect the change. So they can get a hit or a miss, a right answer or a wrong answer. They can lick when they're not supposed to. They get nothing. And they may fail to lick when they're supposed to. Okay? And then, for 25 minutes, they present the animal with no natural images. So these Gabor patterns, stripes, gray screens, white screens. And then for another 60 minutes, they redo the presentation of the same images in the same sequence, except that the lick port has been removed. So the animal cannot respond and cannot be rewarded. Okay? That's the experiment. We're good? So we take this raster plot from the data, like 30 seconds of the data, and let me explain a little bit more detail, something that I didn't mention before when I was talking about urethane, which is a variant of the phenomenological renormalization group which is called momentum space renormalization group. Again, this was introduced by Bill Bialek in that first paper I mentioned, 2017, Serena Brady and Bill Bialek. So you take these eigenvectors of the covariance matrix, so I have n neurons, n by n covariance matrix, and you project, you create projectors to project this activity into a number n cutoff of eigenvectors, and then normalize the activity to some kind of univariance, for instance. And then you do, if you do this, for instance, for this one neuron, if you take all the covariance vectors, you recover the original sequence, and then if you take n over 2, so which n over 2? The ones with the largest eigenvalues. And then n over 4, n equals n over 8, n over 16. So you are coarse graining your data according to significance in this eigenvalue spectra of the covariance matrix. And then when you put them together, all the neurons, you do this for all the neurons in the same time series, and you take the average activity after you have renormalized. What you get is something like this. So here I'm showing this probability of activity in this data set after you have renormalized a couple of times according to this procedure. Okay? So this is momentum space renormalization group, phenomenological renormalization group. And what you get here is, again, something which looks non-Gaussian. Here, shaded, light shaded, you can see the original distributions and how it evolves to this non-Gaussian stuff. Whereas if you come here and shuffle all the spikes, and therefore attempt to destroy correlations, you get something like a Gaussian here, which, by the way, the kurtosis of a Gaussian should be 3, and here it's 6, right? Because you still have some garbage here and there, some irregularities, because you don't manage to shuffle enough in a 30-second window. So here, I interpret this as this window of 30 seconds of data is showing me signs of criticality because the renormalized activity has a non-trivial kurtosis way higher than what you get for the surrogate data, in the spirit of what I mentioned before. But this is not always the case. There are other windows for which you do the same procedure and you get something like that. So kurtosis here is 6.6 for the data and 6.1 for the surrogate data. Is this different enough? So we have to come up with some ideas of how to tell whether it is different or not. Okay? And this happens for some windows, and some, depending on the window you're looking at, you may have this something like that or something like that. Meanwhile, the animal is performing a task in which he gets wrong and right answers here and there to some extent. So the question is, okay, before we go to the question, let me paint a portrait similar to the one which I showed you from Michael Fisher's review on the renormalization group. For the phenomenological renormalization group, what's happening here is the following. So yeah, this is the portrait I referred to in models. Here we don't have models. Here is for a model. So you have a non-trivial fixed point here, and you have trivial fixed points elsewhere. The signature of being here being non-Gaussian. So here, the picture we have is that you have some windows of data for which when you renormalize, when you coarse grain according to this procedure, you eventually approaching this non-Gaussian fixed point, and you can see signatures of non-triviality in the data when you look here. Of course, in order to do the procedure precisely, you have to do infinite times. We don't have infinite data. Whereas for some other windows, they tend to be trivial. They would converge to this Gaussian fixed point here, like this red one. Okay? And so the signature of being here in this trajectory, like all surrogate data do, is to have low kurtosis in the final renormalized distribution. Whereas the signature of being here would be some high kurtosis in the renormalized distribution. The question then being, hello? There you go. How far must one be to be considered critical? So I'm interpreting kurtosis of the renormalized distribution as people have been looking for distance to criticality in this field. I don't have a distance to criticality, but I have a distance from triviality. Okay? How far away I am from being trivial. How far must one be to be considered critical? I mean, is it different enough? So what I'm going to show you now is that I do this over all the windows of all the animals, and I look at the distribution of kurtosis that I obtain overall. So this is what I get. So the data, in general, has, of course, larger kurtosis value than surrogate data. Okay? And which kurtosis is large enough to be considered different from surrogate data and therefore sufficiently robust to have some scaling or something? So it depends on how strict, honest, and welcoming you are. So, for instance, this is the over-enthusiastic gullible. Would put the threshold here, right? Yeah, that's critical enough. This is the z-scorer. That would be me, more or less. That's where I would put myself. Then there's, of course, the skeptic and the denier, okay? So my proposal is that we make, how could that be wrong? Let's try to make everybody happy, right? What could go wrong with that? And just put a knob, and you choose whichever level you want, and let's see what happens. Okay? So what I'm going to do is introduce this kappa threshold. Let kappa threshold be an arbitrary threshold that I'm going to control and show the results as a function of it. Okay? So let's say that I chose this one, and for kappa larger than kappa threshold, a window is deemed to have non-trivial scaling because I so determined. I chose the threshold. I chose that here is large enough that everything above this thing is considered to have scaling. Everything below it has no scaling, yeah?
Before you go too deep into the kurtosis argument, going back, you mentioned that the way you do this rescaling is you take the leading eigenvectors of the covariance matrix and project your data onto it and then compute these histograms.
Yeah, it looks like PCA. Yeah.
Is it PCA?
Yeah, it looks like PCA. It is to some extent, except that in the decreasing order and in how it evolves as you filter out. And I never get to one or two.
Okay, but again, think about
principal
like
components like people usually do. So I have more than that. I have way more than that. Yes, but sorry.
And okay, so it's a little bit like PCA. And so I was wondering if you were not just to measure kurtosis, but the variance explained by the lead. So say you do this n over 16, so you would have like 5 to 10% of the number of recorded units would be the eigenvectors on which you project. How much variance does that capture of the raw data? And if you look at that over time, like just R square of how much your projection.
I don't know the answer, but I'm not sure it will be relevant because you can have variance in the data. What we're trying to do here is not explain the variance. What we're trying to do is extract some signature of non-trivial behavior that based on theory you expect to be of a critical system. So I don't know.
The reason why I'm asking is I have a hunch that there might be a link. If you think about neural dynamic that is very high dimensional and you project it down on these eigenvectors, the projections, when they end up looking, like a high dimensional object is always a sphere, and if you project that down, you end up with something that might be closer to a Gaussian. But if your high dimensional object is lower dimensional, then even the projection might have more structure to it, and you might see this excess kurtosis as more structure to see in the dynamic activity patterns.
So let me see if I understand what you're saying. You're saying that I could get more information or different information.
I think the dynamical state of the neural network might change over time. There might be states when the neural activity is more filling of state space, and so it fills this 128-dimensional space, more like a hairy ball of sorts, and then you project it down with this projection scheme into a much lower dimension, like hyperplane. And if you project this high-dimensional ball onto this plane, you would have some lower dimensional dynamics. It's kind of like light passing through a structure. You see these projections.
Mm-hmm.
And I'm trying to develop more of an intuition of what this geometric object of neural activity does in these different regimes. When you observe excess kurtosis, that seems to be associated with a higher richness of dynamical activity. You have these heavy tails that then appear. And maybe this is linked to a more structured geometric pattern of neural trajectories. And then when you do this projection scheme, you see that excess structure there.
Okay, I got it. I see what you're saying. Yeah. So we didn't do this here, but other people in this community have. So I know Antonio Fontanelli and Woodrow Shu, University of Arkansas, they did that. And results are very interesting. When they do that, they manage to remove some, I would say garbage, but I don't want to. When you lower the dimensionality of the data, then those avalanches that I mentioned here that are troublesome, then they work all right. So my answer is almost unrelated to your question, but in the sense that, yes, there's something to be gained by doing that, perhaps. But we're not doing this here. Yeah. Okay. Is it okay what I'm doing here? So I'm choosing a threshold. I mean, this is a free parameter. I'm going to show everything as a function of that. And then you have the mice performing the data. So here's a 30-second window, another one, and the mice is performing image detection, the change of images. Sometimes he gets a miss, sometimes a hit, hits and misses everywhere. In windows which, according to my criterion, may be deemed critical or non-critical, with non-trivial scaling or without non-trivial scaling. And the question, and of course, we need a control for that as well. So we have this trial shuffle data where we mixed up all the hits and misses so that we can something to compare with, right? Because, by the way, this is important because these mice have been pre-trained for weeks, so they're very good at the task, and they get like 70% of the answers correct. So they're very good. I don't know. So the question is how distance to triviality, kurtosis, impact performance, if at all. So this is the plot. It's a very simple plot, but I'm very fond of it. So here's the hit rate, and this is kurtosis threshold. So say take kurtosis equals 9 here. So whenever I look at the windows which have kurtosis larger than 9, therefore they have scaling according to this criterion, their hit rate is this one. Whenever I look at windows which have kurtosis lower than 9, their hit rate is lower. So the blue curve is above the red curve. That's the main result. Windows with scaling have better performance than windows without scaling, by whichever criterion you happen to prefer. You choose whatever you want to put the bar. You choose where you are on this axis, and wherever you choose to be, scaling windows have better performance than non-scaling windows. And here's the trial shuffle data. It should be where it should be. It basically shouldn't change, right? Okay?
Sorry, maybe I missed it. So what's the average hit rate you said?
It's 0.7 or something. That's the orange line. So if you filter by scaling, so a sign of criticality in my interpretation, then it goes up. Otherwise, it goes down. You can choose more fancy number. So higher kurtosis, better performance. You want to relative performance gain if you want. It's nicer to show. It's the blue, blue minus the red divided by the average or something, right? So it's just how much you gain by being critical. And so you can go all the way to 24% gain. Of course, in something which is very unlikely, by the way, this dashed line is the probability of finding a kurtosis distribution. So yeah, those extreme guys are very unlikely, but when they are here, they do have a higher performance. You can choose whichever global number from here. Take the maximum here, for instance. At peak, where most probable events are, you get like 15% or so, 10%. I don't know. On average, 10%. If you think that 10% is not enough, go talk to Mother Nature and natural selection. So that's the first critical take-home ideas. So non-Gaussian scaling dynamics in the mouse primary visual cortex are closely linked with improved performance in a visual task, supporting the idea that critical dynamics underlie functional advantages in neural processing. Yes?
Have you done a similar analysis with other numbers? For example, plotting the performance as a function of the CV?
Yes. Yes. Then it gives you nothing because CV is usually very low. It's low all the time because the animal is awake and alert. So CV doesn't change much. CV is always low. The animal is paying a lot of attention. Activity desynchronizes. And precisely, my urethane results would predict no signal for criticality. And yet we do find them. So the two asynchronous states
are really different.
Completely different. Which, okay, depends on who you ask. For any electrophysiologist, they say, well, obviously, the animal is anesthetized in one case and not anesthetized in the other case. Right. But people have always looked, to the best of my knowledge, at this asynchronous activity as something, well, it's asynchronous activity. It's that thing that we see everywhere, right? So I find this very interesting, that you can tell apart these two different asynchronous activities with this technique.
And then a second potential plot that one could make is not having the CV on the x-axis, but an estimate of the intrinsic dimensionality of the neural activity that you can also get from PCA. Have you tried that?
That's a good one. I'm going to write it down.
Yeah, it's a Larry Abbott idea. They have a measure for it based on the eigenvalues of the covariance matrix, where you just look at the distribution of eigenvalues.
Okay, great. Okay, I'll look it up.
An intrinsic scale and
to see if there's some kind of correlation, right? Yeah, that's a nice idea.
Yeah. A more richer neural dynamics gives also rise to better performance.
Yeah, good one. Hadn't thought of that.
Thanks, Daniel.
Now, okay, I find this very interesting, but it gets better. Please bear with me. So we have these results for performance correlating with criticality. Now, there are the two other parts of the experiment. Of course, here, I cannot measure performance because there's no performance at all. The rat's doing nothing. It's just sitting there looking at the images, right? But I can look at something else. Here, I'm showing, again, for this active part of the behavior, the cumulative, the fraction of windows above threshold. So it's like the cumulative distribution of kurtosis, okay? In blue here, for this part, I'm showing the cumulative distribution of kurtosis. Well, I can measure kurtosis here, even though I cannot relate with performance. I can measure kurtosis here, and when I do this here, I get this cumulative distribution of kurtosis, which is hard to compare when they're separated like this, but if I join them like this, you can see. Ta-da! There you go. Here, in the passive phase, kurtosis get closer to surrogate value. There's no need to perform here. There's nothing to do here. Your kurtosis go down. There's no need to be critical here. Yes, Italo? You need a microphone?
Do you
That's a good point. Possibly. I would argue this supports the idea that it is useful for behavior. And therefore, if you're not behaving, or in this case, you're not performing a task, there's no need to be there. Whether it's more costful or not, I don't know. But the pressure wouldn't be there, okay? And you can measure here as well. I don't remember if the animal has ever seen these non-natural images, actually. But they certainly had not been trained to get rewarded by these images. So when you put them here and calculate the kurtosis, then the kurtosis gets even closer to trivial values here, okay? And this is, so signals of criticality, if you want, are modulated by task involvement. How much involved the animal is in the task. I wanted to use engagement, but then someone criticized me because engagement is something else in biology. You cannot use it that easily, apparently. I don't know. I would prefer engagement. Is it okay to use engagement? You biologists, you would be fine with that? Yeah. So the mouse brain's ability to self-regulate and display scaling dynamics predominantly while performing a task reinforces the idea that scaling plays a role in enhancing sensory processing. Okay? So this, oh, and you can look at exponents for the physicists in the room. The exponents are different in the three phases. There's a group data, and they tend to go, these dashed lines are trivial values of these exponents. So static exponents alpha and beta shift closer to trivial values as engagement decreases. So I reverse the order here. Active, passive, and non-natural images. And they go in a different direction with the dynamic exponent z, it has to do with the autocorrelation time. I don't know how to interpret this. And I don't care that much, actually. To the what? To the urethane data? Yeah, they're more or less. Yeah, more or less, yeah. I'm trying not to spoil myself. No, I think the papers on archive. Forget about this for a while, and we have data from the Human Connectome project on fMRI data, like 700 patients or so. We do this thing, and the exponents, the three exponents, fall into a line in 3 dimensions, so they're not independent. So it's more about scaling relations there. So we don't know. So this paper is still under revision here, and I would love to run this thing on other data. So Ahmed was telling me about the data about the model he has on this collective agents foraging. Yeah, we'll gladly run this thing. We're running this same techniques in other neuronal data, and we find no signature of criticality. So that's to answer her question, right? What if it's not there? Yeah, sometimes it's not there. And we try to interpret this. We're using this data from the LA Institute, going away from the primary visual cortex. They have all the regions. As you distance yourself from V1, the signatures get weaker, as perhaps they should, I don't know. So you see, we're refining this, what started as a very rough idea, a critical brain, well, the critical systems that are engaging in this task, perhaps, right? So just want to thank everybody that somehow collaborated in whatever I talked about yesterday and today. This is my group. Look, we're smiling here. We're a happy bunch, okay? So Philippe is there. And people who paid for everything. Thank you very much. I'll take your questions.
Okay, questions?
So he gave this really nice idea that staying at criticality costs energy. Would it be possible to run an experiment where you have like calorie-deprived mice and see where they perform worse in the criticality department? Sorry. So he gave this great idea that
Yes.
criticality might be energy expensive.
Yes.
So would it be possible to have some, I don't know, calorie-deprived mice performing these tasks and see if they are less able to stay close to criticality?
It is possible, but in the scale of the Allen Institute, only they can do this, I think, yes. But yes, so given that we find this context-dependent signal for criticality, then the doors are open to test changes in context, however we want to play with. But yeah.
I would argue that it would be the opposite, because in calorie deprivation you have increase in cortisol, and this prioritizes seeking behavior rather than homeostasis behavior. So probably the organism will change to energy expenditure to be more efficient in order to overcome calorie deficiency.
So, Mauro, I have a question. So, because everything about criticality is actually a kind of indirect evidence. So, for example, in these last parts of your talk, it was about the kurtosis, and you interpreted this as a sign of criticality. But, for example, I think another type of interpretation is going a little bit in the direction of Manuel. It could be that actually what you are measuring is really the synchronicity, right? That could be obviously related, and it's not something that is contrary to your hypothesis, but maybe the criticality is not necessary to explain the same kind of phenomenon.
Could be. Yeah, I'm open to that. But then we will have to find some kind of data set and or model that will give all those signatures, because it's not only kurtosis, they're the exponents, and whatnot, right? So right now I have a student in my lab doing PRG on models. So the model, we know where the critical point is, we know everything about the model, and then it can run this thing and check whether it is true that those signatures appear only there. And it is, so far, for the couple of models that we have studied. I agree. And I would counter that. So what would a direct evidence be of criticality? I don't know. I don't know. It's very hard, right? So again, it's just a hypothesis that people thought could be useful. And I think it has given some interesting fruits. So, but you're right. Yeah, it is indirect, yeah. By the way, synchronization in this data, I find it very, I don't know where I would find it. Perhaps after I throw everything and project into lower dimension, maybe I can find something there. I'm not sure. We can have a look, yeah. It's very asynchronous behavior. The animal is very alert. Oh, and going back to one of your questions, people have tried to correlate with like pupil diameter, stuff like this, and it works more or less, not much, for this data set. For others, it works well.
Okay. Another question? Okay, so today we will end a little bit earlier than the usual. Professor Mauro has to leave soon, so let's thank him again.
Thank you very much.
Okay, everyone. So today we are